is Biomed Cafe menu. Golgi Apparatus will be presented today. We will talk about Golgi Discovery, Golgi Structure, Golgi Function, quiz game and puzzle. I'm Dr. Biomed, cellular and molecular biologist. Hi everybody, welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified and summarized form. Golgi Discovery, they saw something others didn't see. The Golgi apparatus was observed in 1897 by Camillo Golgi. While examining neurons that Golgi stained, he identified an internal reticular apparatus. This structure became known as the Golgi apparatus. In the 1950s, when the electron microscope came into use, the existence of the Golgi apparatus was confirmed. Golgi characteristics: is membrane-bound organelle of eukaryotic cells. It moves molecules from ER to their destination. Is located in the cytoplasm next to the ER. While many types of cell contain only one or several Golgi apparatus, plant cells can contain hundreds. It also modifies products of the ER to their final form. It's also called the Golgi complex or Golgi body. Is comprised of a series of flattened sacs that bud off the ER. Golgi is slightly smaller and smoother in appearance than ER. Golgi structure. Constituent unit of Golgi is cisternae. It also called dictyosomes. Cisternae are flattened sacs that are stacked on each other, much like the ER. A mammalian cell typically contains 40 to 100 stacks of cisternae. Cisternae have two parts in their structure: membrane and cisternal space or lumen. Membrane is a phospholipid membrane and encloses the lumen. It separates from the cytosol. Cisternal space or lumen is internal space of cisterna. Between 4 and 8 cisternae are usually present in a stack. The collection of cisternae is broken down into 3 compartments. Cis is closest to the ER, medial is middle of cis and trans is the site furthest from the nucleus, which secretes vesicles to various parts of the cell. Compartments making up 2 main networks: the cis-Golgi network or CGN. The CGN is the first cisternal structure and associated vesicles, and the trans-Golgi network or TGN. The TGN is the last cisternal structure and associated vesicles, which proteins and lipids are packaged. Models of vesicular trafficking. There are so many models, but here we'll talk about 3 models. The first model is the cisternal maturation model. It's the most accepted model among scientists. Cisternae themselves tend to move from the cis phase to the trans phase of the Golgi over time. These cisternae age as they move towards the trans phase of the Golgi and their product becomes fully mature. Secretory vesicles are transported in an anterograde direction along with cisternae maturation from cis to trans of the Golgi. Golgi resident vesicles are transported molecules in an anterograde direction back to the ER. The second model is rapid partitioning model. The Golgi stacks are distributed into two subdomains: processing domain and exporting domain. Secretory vesicles are concentrated at export domain, but Golgi resident vesicles are concentrated in processing domain. The third model is Golgi tubule-mediated transport model. Each cisterna within a stack is connected by Golgi tubule. Golgi tubules are involved in the rapid bidirectional transport within the Golgi complex. Rapid bidirectional transport is fast anterograde traffic of secretory vesicles and the retrograde traffic of Golgi resident vesicles. Golgi functions. Number 1, modification. Number 2, packaging. Number 3, sequestration or exporting or transport of cargoes. And number 4, organelle producing. Modification in Golgi complex is as phosphorylation, removal of mannose, glycosylation, sulfation, and addition of fatty acids. Phosphorylation. Phosphorylation is adding a phosphate group into the protein by kinase. It occurs in the cis. This modification usually affects cell processes related to the cell growth and cell signaling. Removal of mannose. Removal of mannose occurs by manocytase in the cis and medial cisternae. Glycosylation. There are two glycosylation: N-link glycosylation that is occurs in the RER and O-link glycosylation that it occurs in the Golgi complex. Now we want to talk about O-link glycosylation. N-acetyl glucosamine, galactose, fructose, and sialic acid create an oligosaccharide. This oligosaccharide attached to the oxygen of the serine or threonine of a protein by GalNAc transferase. It occurs in medial cisternae. Sulfation. Sulfation is adding sulfate onto tyrosine of proteins by tyrosyl protein sulfur transferase. It occurs in the trans cisternae. Addition of fatty acids. Adding fatty acids onto cysteine of proteins is done by ZDHHC enzymes. This makes the protein more hydrophobic and is very useful for proteins that are parts of membranes. Another function of Golgi complex is packaging. Golgi can be similar to a post office. After modifications, Golgi packaged the products in vesicles and tagged with markers that indicate where the vesicles need to end up. It occurs in the Trans-Golgi network or TGN. Tag or label is different depending on the destination. Destination can be a plasma membrane or the cell exterior or lysosome. When the destination is plasma membrane, vesicle name is membrane vesicle. Vesicle type is clotting-coated vesicle. Vesicle content is membrane proteins and lipids. There is no tag. One example of membrane vesicles is vesicles containing phospholipid, internal and peripheral proteins. When the destination is the cell exterior, vesicle name can be exocytotic vesicles or constitutive vesicles. Vesicle type is clotting-coated vesicle, and vesicle content is secreting proteins. Label or tag is oligosaccharide. One example of exocytotic vesicles is vesicle containing antibody released by activated plasma B cells. After packaging, the vesicles bud off and immediately move towards the plasma membrane, where they fuse and release the contents into the extracellular space. When the destination is the cell exterior, vesicle name can be secretory vesicles or regulated vesicles too. Vesicle type is clotting-coated vesicle, and vesicle content is secreting proteins. Label or tag is oligosaccharide. One example of secretory vesicles is vesicle containing neurotransmitter released from neurons. After packaging, the vesicles bud off and are stored in the cell until a signal is given from their release. When the destination is lysosome, the vesicle name is lysosomal vesicle. Vesicle type is clotting-coated vesicle. Vesicle content is lysosomal enzymes. Label or tag is mannose 6-phosphate or M6P. Adding M6P occurs in the CGN and budding occurs in TGN. One example of lysosomal vesicle is vesicle containing digestive proteases. The vesicle first fuses with the late endosome, and the contents are then transferred to the lysosome. Another function of Golgi complex is secretion or exporting or transport of cargoes. After packaging and labeling, the vesicle is excreted from the Golgi on its way to its final destination. This occurs in the trans-Golgi network or TGN. Vesicles are carried to its destination within the cell by the cytoskeleton. Another Golgi function is organelle producing. At first, cell membrane. The Golgi and ER work together to produce cell membrane, as well as recycle the cells of vesicle. Lysosome. The Golgi creates lysosomes. Plant cell wall. The Golgi and ER work together to produce plant cell wall. And acrosome. The acrosome is an organelle that develops over the front half of the head in the sperm cells. It's a cap-like structure derived from the Golgi. Key points of Golgi. In eukaryotic cells, the Golgi apparatus is the manufacturing and shipping center of the cell. A Golgi complex contains cisternae. Cisternae have lumen and membrane to separate them from the cytoplasm of the cell. The Golgi complex is capable of both disassembly and reassembly during mitosis. The Golgi complex is capable of both disassembly and reassembly during mitosis. While the ER produce most of the products, it's the Golgi that is responsible for the modification, packaging, and exporting them. Thank you for watching. Please subscribe to this channel, and if you like the contents of this video, please like it. Now it's time to the quiz game and puzzle. I have put the link to the quiz game and puzzle in the description of this video. Download it for free and enjoy it. Until the next video, bye!